So I thought before we delve into the details of protein synthesis, I'd give you a good sort of conceptual understanding of what has to happen by providing you with an overview. So first of all, I'm hoping that you understood the beetle and Tatum information and that experiment made sense. If it didn't, that's okay, because the main piece of information that I need you to remember from the beetle and Tatum experiment is that the function of DNA is to code for proteins. The beetle and Tatum experiment itself identified that there was a relationship between genes and enzymes, in that one gene codes for one enzyme, and that was extended to apply to all polypeptides. Hence, the blueprint for all proteins is contained in DNA. That's what DNA does. It codes for proteins. You would have learned from, I want to say maybe grade 8, possibly grade 10 science, that proteins are made on the ribosome. The ribosome is responsible for making proteins, right? And the ribosomes are located in the cytoplasm. So if I were to draw just a quick rough sketch of, a cell, right, I would say that here's my nucleus, and my DNA is contained in my nucleus. And then somewhere around here I have my endoplasmic reticulum. I'm really terrible at this. And then surrounding the endoplasmic reticulum, I might find some ribosomes. So the problem is, in protein synthesis, this is the problem that arises, is that our DNA is stuck in the nucleus and cannot leave. However, the ribosomes, which is where proteins are made, are located in the cytoplasm. Well, DNA is what tells us what the code is for making the proteins on the ribosomes, but the DNA can't leave. So that's a problem. That is a problem that protein synthesis has to somehow overcome. And so it does so by a couple of different processes. And so I'm going to go into detail about that. Before we move on with the note, I want to talk to you about the fact that, RNA is another nucleic acid that becomes really important in protein synthesis. And we're going to talk about 3 different types of RNA. Okay, so here we go. 3 different types of RNA in today's lesson. So the first one is mRNA. And the M in this case stands for messenger. We're going to learn what messenger RNA does. The second one is tRNA. And the T in this case stands for transfer. So we're going to find out what tRNA does. And then the last one is called rRNA. And the R stands for ribosomal RNA. So by the end of today's lesson, it's not escaping me that this last one's in green, by the way, which is really bothering me, but let's move on. So it's, in today's lesson, I'm hoping to introduce you to each of these types of RNA and to highlight their involvement in the process of protein synthesis. However, in future lessons, we're going to be delving a little bit deeper into protein synthesis, and they will come up again. So let's first talk about messenger RNA, or mRNA. So I like to think of mRNA as being, I like to think of DNA as sort of being like Cinderella, and Cinderella, is stuck at home and can't leave her stepmother's castle, right? Well, what does she have to do? She has to transform using the help of her fairy godmother to be able to leave her stepmother's house. So in the same way, mRNA is basically just a transformed version of DNA that is able to leave the nucleus. So what mRNA does and what its role is to make a copy of DNA, but in a single-stranded mRNA version. It's going to make a copy of the gene that needs to code for a specific protein. And that mRNA is therefore going to be able to leave the nucleus and move to the ribosome. So that solves the problem of the fact that DNA is coding for these proteins, but the DNA can't leave to get to the ribosome. So this whole process of mRNA making a copy of the DNA is called transcription. So let's look at how the names of these, it's super handy actually, because the names of these processes and structures help us to know what they're doing. So what does a messenger do? A messenger sends a message. So the mRNA, messenger RNA, is just sending a message to the protein about what the genetic information is. And that process of copying DNA is called transcription because, well, what do you do when you transcribe information? You're basically copying information. Okay, so phase 1 of protein synthesis involves mRNA making a copy of the gene, making a copy of DNA, but in mRNA form, and then leaving to go to the ribosome. That is called, the copying process is called transcription. But then eventually that mRNA is going to leave and go to the ribosome. It's going to exit the nucleus and travel to that ribosome. And then what it's going to do is translate DNA's code into a protein. And that process is called translation. Okay? So 2 steps to protein synthesis: transcription and translation. So let's look at translation, just the overview of translation, right? So here what we have, if you can imagine, we have here this blue little segment here is representing that mRNA strand, that copied version of DNA that can exit the nucleus, and it's arriving at the ribosome. So I can't remember, I feel like at some point in our course we've already talked about how the ribosome almost looks like a hamburger, like the bottom bun and the top bun, and they basically clamp on to the mRNA. So you can sort of think of this, the ribosome is clamping onto the mRNA and then sort of reading along. And it's going to read the code or translate the code in the mRNA into the amino acids that are going to eventually form the protein. So that is basically what translation is happening during translation. Okay, so the ribosome is going to be able to translate the RNA nucleotide message into certain amino acids, and then that, you're going to end up with a chain of amino acids that become a polypeptide and then a protein. So essentially this is, if we had to kind of sum it up in 3 main steps, we would say the DNA becomes RNA and the RNA becomes protein. And this is what is referred to, and it's a big deal in biology, it's called the Central Dogma of molecular biology. So the central dogma, a dogma is like a truth or an understanding. So that central dogma is that DNA becomes RNA, which becomes protein. That's it. But the process by which that happens, we can say, all right, well, DNA to RNA, that process is called, what is it called? Transcription. And then RNA to protein, that change involves translation. So because our mRNA is going to be sending that message from DNA to the ribosome, it's pretty critical that we understand the differences between the structural components of DNA and RNA, because DNA and RNA do have some subtle differences that are important to note. The first being that they have a different sugar. Now that doesn't really play too much of a part in protein synthesis at this level, but it is worth noting. So DNA has deoxyribose. Still, they're both 5-carbon sugars, but ribose, which is unique to RNA, has a hydroxyl group here. So notice the deoxy, meaning suggesting less oxygen, and we see that here with the substitution of hydrogen and hydroxyl. Now, in previous lessons, we've looked at the differences between DNA and RNA in terms of their nitrogenous bases, whereas DNA has adenine and thymine pairing up, and cytosine and guanine pairing up. We know that in RNA, the uracil replaces thymine, right? So uracil is going to pair with adenine instead of thymine pairing with adenine. So that's important. And if we compare uracil and thymine, we can see that they are structurally similar. They're both single-ringed pyrimidines, but there's a difference of a methyl group on our thymine, and whereas there's just a hydrogen on our uracil. But they're similar enough so that they can still both form those two hydrogen bonds with our adenine purine, right? So uracil substituted for thymine in RNA. So in other words, if we had, let me kind of try to make this visual for you. Let's say in DNA, along the whole strand, I had, adenine, then I had thymine, then I had cytosine, and I had guanine. What that would mean when mRNA is trying to make a copy is that the copy would have the complementary bases. So adenine normally, if the complementary strand were DNA, would have a thymine here, but instead it has a uracil when I'm making mRNA. And so on and so forth. So thymine still pairs with adenine, cytosine will still pair with guanine, so the complementary strand will still be complementary. It's just that any time that I see adenine on my DNA, I'm going to have my mRNA version have a uracil instead of a thymine. So, moving right along, we know that our DNA is double-stranded, so it should make sense to you that just like with DNA replication, that the DNA is somehow going to, the two strands are going to have to somehow become separated if we're going to make a copy of it. And an RNA, the RNA that leaves the nucleus is only going to be single-stranded, right? So here's our single strand. But we'll get into those details tomorrow. Now, I promised you we'd get to it, and now we're there. So transfer RNA, okay, well, what is something that is transferring doing? It's possibly bringing something from one place to another. And that's exactly what tRNA's role is. Its role is in the translation phase of protein synthesis. So right here I have, let's just imagine that this is my mRNA strand. And then possibly right here is a ribosome. And you'll remember that the mRNA is basically getting read by the two halves of our ribosome hamburger bun. It's not called that. I just added that in. But then somehow there's a code on this mRNA strand that our ribosome is going to read and somehow translate into amino acids. But those amino acids don't appear out of nowhere. It's actually the tRNA that is responsible for transferring or bringing the amino acids in to build the protein. Okay? So mRNA arrives at the ribosome. The ribosome clamps on the mRNA and somehow reads its code, which tells our tRNA which amino acid to bring in to start building the protein. Again, that is a complicated process, which we will be looking at in its own unique lesson, and that's coming up shortly. Now, the third type of RNA is rRNA, which I mentioned stands for ribosomal RNA. And ribosomal RNA is just RNA that actually just makes up part of the ribosome. Right here we can see the hamburger bun that I was talking about, upper and lower, okay, so larger and smaller subunits. But within that ribosome, we will see some RNA as part of the structure. All right, so I want to depart from the note for a couple seconds here because I'm thinking this is usually the part in the lesson where students are like, well, the mRNAs come in, there's the ribosome, two components, and I've got my tRNA that's coming in and bringing in the amino acid. But, first of all, how the heck does this ribosome read code along this mRNA strand? Like, what is the code and what is that all about, right? Well, it may have occurred to you that it might have something to do, well, what makes, one nucleotide different than another nucleotide? They both have, all nucleotides are going to have a phosphate. All nucleotides are going to have our some kind of 5-carbon sugar. But what they have, what each nucleotide has that potentially is different, is a different nitrogenous base on it, right? And in DNA we have 4 of them, and in RNA we have 4 of them, and some of them overlap. So it turns out that the code that our ribosome is reading has to do with the order or the sequence of those nitrogenous bases that we find on our mRNA strand. Let's get to that. Before we start writing out the note pieces, the words that go with this, let's just take a look at this diagram, which I think kind of gives you a good overview of the pieces and the reading of the code, right? So, and I really like a lot of the details that are here that will remind you of some of the details we saw in DNA replication, because there are some similarities. So imagine that here I have, my DNA, my double helix has unwound, and the 2 strands have separated so that only a specific gene can be copied, which is just a certain segment of the DNA strand. So, and remember that if DNA is coding for protein, it's not going to copy the entire chromosome. It's only going to copy the small section or the small gene that is coding for that protein. So imagine that I have my DNA strand that has opened, and this is the section, this is the gene that needs to be copied. Now notice that it's in the 3' to 5' direction. And so remember, this DNA can't leave the nucleus. It's got the gene information, but it can't leave the nucleus. So it makes a copy of itself, but in the form of our mRNA, right? And that process is transcription. So in transcribing from DNA to mRNA, notice that the strand runs anti-parallel. Not super surprising. But also notice that it is complementary. So my C pairs with G. A would normally pair with T in a DNA strand, but I've subbed in uracil because, yes, this is mRNA. And on and on and on, right? So in here lies the same complementary sequence as I saw in my DNA strand in the form of mRNA. But the problem is that, well, I want you to think about how many amino acids. Take a minute to think about how many amino acids we have. I'm hoping you remembered that we have 20 amino acids. But how many nitrogenous bases do we have? Well, whether we're talking about DNA with thymine or RNA with uracil, we still only have 4 nitrogenous bases. So how do we take 4 nitrogenous bases and get 20 amino acids out of them? Well, we think about making different combinations of nitrogenous bases that together code for a particular amino acid. So for example, and by the way, we call those combinations codons. So what is a codon? A codon is a group of 1, 2, 3 amino acids in a particular sequence that code for a particular amino acid. So for example, the codon GUU indicates the amino acid valine. The next sequence, GCC, is a code that tells the ribosome, tells the tRNA to bring in the amino acid alanine, and so on and so forth. And so scientists have developed, through experimentation, have developed a nice handy chart that allows us to identify what codon is associated with which amino acid. And I'm going to take a look at that right now, and then we'll come back and write some stuff down. So here's the chart that I was talking about, and you should be able to find this in the back of your textbook. I don't know the page number off the top of my head, but you should be able to find it in that appendix section, or possibly in the chapter section where you'll find the overview of protein synthesis. So this is how this works. I'm just going to, arbitrarily make up some kind of a codon. Okay, let's just say I have ACG. Just totally made that up. I want to know, well, if I find that on my mRNA strand, what is that actually going to represent in terms of amino acids? So which amino acid can I expect my tRNA to bring in? So I would look at the first base. Well, the first base in this is an A. So that means I'm going to start first base, I'm going to start in this row. Then my next base is a C. So then I say, well, second or middle base is a C. So now I know I have to look at this particular column. And then I have to look at my third base, and the third base I'm going to look at is a G. So third base, I go down and it is a G. So then I can go over and see ACG is THR. And THR, I'm not going to lie, I had to pause this video and go look it up, stands for the amino acid 3-onine. So there we have it. So that is how scientists over time developed and created this chart that helps us to identify what the mRNA is trying to tell us about what amino acid tRNA ought to bring in. I think we're at the point where we're ready to write a little bit of this down. So let's go through this. I'm just going to kind of go bullet by bullet, I think. So during transcription, the chemical language for DNA and RNA is the same. We have those nucleotides, right? We can see that we have nucleotide, nucleotide, nucleotide. During translation, the base pair language is converted into amino acids. And how does it do that? Well, even though we have 20 amino acids, we have 4 nitrogenous bases. The 4 bases are going to be put into different sort of combinations in triplets specifically, and those triplets are called codons. Okay, so DNA makes a copy of itself in the form of mRNA in a process called transcription. The mRNA will look a lot like the complementary strand of DNA except uracil. And then we make groups of 3 in sequence along the chain, and each of those groups of 3 nucleotides gives us what we call a codon, and that codon can be translated into a specific amino acid. And so one after the other, we have those amino acids in the proper sequence so that we produce, so that we have the right protein. Phew. Well, this next sentence tends to confuse students, so I am going to have you read it through with you. You're going to pause the video and then think about it, and then I'm going to show you, I'm going to go back to that chart, and we're going to try to work through it. So it says most amino acids have more than one codon. Okay, we call that redundancy, but each codon represents only one amino acid, so it's non-ambiguous. So let me explain what that means. So remember this guy, I'm just going to highlight, and arbitrarily I'm going to highlight this square right here. So this is what we mean by the difference between redundancy and non-ambiguity. So when I look at this, you'll see that CGU codes for arginine, but so does CGC, CGA, CGG. They all code for arginine. So that's what we mean by it is redundant. Okay, multiple codons code for the same amino acid. That just gives mRNA a little bit of flexibility in how it creates its code, right? But I know, and this is what we mean by non-ambiguous, that a single codon can only bring in one amino acid. So for instance, if I said to you CGU, that can only bring in arginine and no other amino acid. Even though there are multiple codons for arginine, each of these codons can only bring in one specific amino acid. That is what we mean by it's non-ambiguous, because that would be confusing for our poor ribosome if we had CGU coding for different amino acids. It'd be like, well, am I bringing in arginine or am I bringing in this other amino acid, right? So the fact that a single codon can only bring in one particular amino acid is really important in terms of not producing, defects in our resulting protein. So you may want to write a little bit down about that just to remind yourself, because these ideas are a little bit confusing. Okay, so glycine, I didn't use glycine, I used arginine, but same idea, has 4 codons, 1, 2, 3, 4. So mRNA has some flexibility, right? But no other amino acid is going to have those codons. And that just reduces error. Gives mRNA flexibility, but it means that the protein that results is going to have far fewer errors or ambiguity associated with it. A couple other points to mention. One of them is that there's always what we call an initiation codon. So, and what that does is it's like, you have your mRNA strand, and it's coming and it's on its way to the ribosome, boom, and it's going to make its way to the ribosome. But what every single mRNA it has the codon AUG. And that basically tells the ribosome, okay, let's start translating this transcription into a protein. Okay, so it basically signals that translation should start. That's it. Okay, so that's what, and it's always, always, always the codon AUG, which always, always, always brings in the amino acid methionine, or MET. So that's one point. The second point I wanted to make was that there has to be a way for this mRNA strand or to signal to the ribosome that translation is done, right? That it's all done. And so the way that it does that is by using a termination codon. Okay, so a termination codon, and there are 3 of them. 1, 2, 3. And so these, unlike our initiation codon, our termination codons do not translate into an amino acid. tRNA is not going to bring in an additional amino acid. It's just going to stop the whole process. And what that does, that termination codon, is it signals to the ribosome, okay, it's time to stop translating and unclamp that, unclamp our mRNA so that its components can be recycled, and to also release that polypeptide that's been building. So, in fact, and this, I apologize, this is getting a little bit messy at this point, but if we look for those stop codons, we can find them. So stop, oops, try that again, stop, stop, and stop. There they are. So you can see that, the word stops there, which is helpful in reminding you. All right, so one last exercise just to make sure you're on the right track. So I want you to try this example. So let's just say, I'm going to say that this is a DNA strand in the 3 to 5 direction. And I want you to tell me what the mRNA polypeptide is going to look like. So take a minute to complete that portion. Pause the video and we'll come back in a second. So if we're looking at the strands, the mRNA that's going to leave the nucleus, then we end up with this. Now, the only thing that, a couple of things I want to mention here. I know that the complementary mRNA strand would be 5' to 3' because they have to be anti-parallel. Another thing that I'm hoping, because I think we know, I've seen your quizzes, you know how to create a complementary DNA strand, but when we're creating a complementary mRNA strand, we need to make sure that at every case where there's an adenine, that instead we're pairing that with a uracil instead. Now, even though DNA might have a thymine, that means that we can still use adenine in mRNA. It's just the case where we have an adenine on DNA that we have to replace what we would have as a thymine with our uracil. Okay, and that happens along the chain. Now what I want you to do is I want you to pause the video again, and I want you to fill in, well, what will the sequence, that primary protein or polypeptide sequence, be for the protein that we're trying to make, given the initial DNA and then our following mRNA strand. Okay, so in order to get our polypeptide, I find it most easy to kind of go 1, 2, 3, and then create like a bar. 1, 2, 3, bar, 1, 2, 3, bar. And what that does is that lets me separate these separate my nucleotides into codons. And then it's easier to reference when I go and I look at my chart. And so AUG, that's a good one to just remember as being methionine, our start codon. So we're going to, so there we are, start. But then as we go, let's see how you did. So methionine, CAU is histidine, I believe. Serine, leucine, and then TRP, which is maybe not right. Okay, no alarm here, people. The problem is, I figured out the problem. This is not a stop codon. So there would need to be a stop codon if that were indeed the end of the sequence. So that's it. Hopefully you did okay with that. And so look forward in the next couple of days for us to take a much deeper look at translation and then transcription. As always, please reach out with any points of clarification, and I'll be happy to help.